basic science has unraveled numerous mechanisms for the neurotoxicity. The primate work has given us perhaps a window into which domains of development may be more affected. And the huge clinical studies have perhaps provided us with some reassurance that brief doses don't lead to significant cognitive decline. Perhaps we are beginning to understand which particular domains of neurodevelopment may or may not be affected. Of course, everyone wants to answer the question. What the question really is, how do we give them, or how do we know the way we give them produces no significant injury? And if there is a risk of injury, then which children are the ones that are most at risk?
Preclinical studies in the non-human primate models have given us significant insight into this problem, and there are a number of questions that could be addressed in non-human primate and other preclinical models as well.
For preclinical studies, we need a better understanding of the neurotoxicity which we observe in the lab. Without understanding the mechanisms, it's difficult to know how to prevent it, and it's difficult to know which children will be at risk or what are the likely phenotypes that we're going to see clinically.
In preclinical studies, early in life anesthesia exposure causes an acute injury of the developing brain. And more recently, we found in our studies in non-human primates that there's also evidence for long-lasting alterations in structure in the brain. So these alterations are sharing similarities with those of brain injuries of other causes, and they are of small intensity, however they are significant. In addition, it appears that early in life anesthesia exposure causes alterations in behavior as the animals grow or the exposed individuals grow. And at 2 years of age, which corresponds to a human child of 6 to 8 years, these animals show increased anxiety-related behaviors. And in our studies, we found that at this age, if we just observe the animals in their social environment, they show alterations in their social behavior. They show decreased social interaction and behavior. In contrast, there's less evidence for alterations or impairments in cognitive function caused by early in life anesthesia exposure. Importantly, these findings in preclinical studies share similarities with those from recent reports in children that found that there's no difference between exposed and unexposed children in general intelligence, in IQ, but parents and caregivers of those children report that they show alterations in their behavior if they just observe them at school or at home. In contrast, there's no evidence or little evidence for cognitive alterations. So it seems to be crystallizing out that early in life anesthesia exposure does not impact cognitive function, but that it affects the behavioral development of the individuals as they grow. And especially if they are observed and observed in their natural or home environment. So one of the major questions in the field that is still remaining is, what is this behavioral phenotype caused by early in life anesthesia exposure? What should we measure in these children? How should future studies be designed to identify this behavioral phenotype? In addition, in most clinical scenarios, the children that receive anesthesia are also undergoing surgical interventions, and many of them that need early in life anesthesia exposure have pre-existing medical conditions. So another remaining question is, what exactly is the impact of the anesthetic alone, of the anesthetic itself?
Things don't happen without money. If there's no money to support studies, the studies don't get done. I do think that there is a real need for continued and increased funding in this area so that it will support the studies. And I mean, just to be perfectly honest, a lot of scientists, they have to be funded in order to do work. If there's no funding available in a given scientific area, no matter what the interest or importance of it is, they're not going to be able to pursue it. So I think it is important for funding agencies to be able to continue to signal that they will invest in this kind of research to attract good people into the field and to support the actual work that needs to be done, which in many cases is neither easy nor cheap.
The non-human primate model is of high value to the field and perhaps the only way to get answers to these remaining questions in the field. However, studies in non-human primates are very expensive, especially as compared to small animal models. One has to consider a substantial number of animals per group to be able to really analyze behaviors. Also needs to factor in potential gender-related differences. And if one aims to follow up these animals throughout childhood into adolescence, into adulthood, this translates into several years of observational studies and animal care. So yes, we urgently need money to facilitate those studies in non-human primates, exposing a sufficiently high number of infants to anesthetics or anesthetics plus surgical intervention, and to follow up these animals throughout childhood, teenage life, into adulthood, to investigate this behavioral phenotype caused by early in life anesthesia exposure.
We've learned a lot, but that there's still a lot that we need to learn about genomics and how we actually integrate that knowledge into clinical practice. And that's been well over 20 years now. So I think we're doing fine. I think we're making good progress, but these kinds of questions are difficult. You can't do the definitive randomized trial, and it just takes time to do the kind of careful studies that are necessary to answer questions like that. So I actually think we're making excellent progress in the field.
The fact of the matter is that if we consider proving beyond reasonable doubt that the phenomenon exists in humans is what we need in order to move forward, that may not be able to prove regardless of the amount of funding. If, however, we believe that the evidence that we have so far in over 600 publications in all the species that people studied is enough to move the field in the right direction, then the funding should be put in the places where it matters. And in my mind, in my opinion, that is actually to focus on the development of new anesthetics that are safer to be used in young kids who need good anesthesia more than anybody else. That is expensive, and that's why the funding is so important. But for us to say that the phenomenon does not exist because we cannot prove it in humans beyond reasonable doubt is hampering us in developing new anesthetics that are most likely on the horizon and important to focus on as soon as we possibly can. That is our duty to our youngest patient population that needs us the most.